down somewhere in your notes, somewhere where it'll be very important. We'll talk a little bit about these dates. And then Amanda, you're in charge of like the Facebook page. Make sure you put this on there. I'll put an announcement out tonight too. So let's first talk about Thursday, August 1st, which is a week from Thursday. I can't believe we're almost talking about August. That's sad. I mean, summer's almost over. Thursday, August 1st. We're going to call this our, look at this, our last lecture. You might say, what? I'll explain. Thursday, August 1st is our last lecture. On Thursday, August 8th, and don't write this down, I'm having some eye surgery. I just found this out last weekend. So, it's completely, don't write that down. But I have to alter the schedule a little bit. So, our last lecture will be Thursday, August 1st. Now, on Thursday, August 1st, what I'm going to do is I'm going to, look at this, update the final exam. So what does this mean? I expect loud clapping after I say this. Not yet.
Oh, I'm ready.
Not yet, Sam. The test that you just took from me will be your last test. So,
that was in class.
So, the test you just took from me will be your last test. So that means that on Thursday, August 1st, after our last lecture, I'm going to add about 10 more questions dealing with the most recent material. So over the this week and next week, I had about 10 more questions to your final exam. So your entire, and this has never happened in the 27 years of me teaching, your entire final exam is take home. And don't tell people this, because this is like probably against some rule. You know, that's not the way I roll, but I have to make some modifications. So in the past, ready, the average grade on the final was probably like an 84, because people got like a 95 on the take home and like a 75 on the normal test. Now, the entire test is take home, which means the average should be in the 90s. It better be in the 90s.
Yeah.
So that means
Now I understand.
now everyone's clapping, right? Thank you. All right, so we're not writing down
Listen, right away.
We're not writing down surgery. We're saying Thursday, August 8th is no class. So that's what you're writing down. So if your brother Omkar is your friend, you know who he is, and he's not here. Of course, I'll explain this again on Thursday, but let's share the news, okay? On Tuesday, August 6th, right, there is no lecture, because remember, last lecture is the first. No lecture, but all sections have lab final. And, look at this, take homes is due. So Tuesday, August 6th is a big day. You're going to come take a lab test, and you're going to submit your take-home exam, which exists right now, which I hope all of you are halfway finished already. And then remember, this take-home now includes an updated version that I will post on the Thursday before. So if you are really up on your game, you're going to have the comprehensive done by August 1st. And then all you're going to have left to do that weekend is the updated 10 or 12 questions or so, whatever I add. And then you're going to turn that in on Tuesday when you come take your lab test. Does everybody get this? Everybody understand it? Yes,
Will
Joel?
you add the 10 questions in Blackboard?
Yeah, I'll just add it to the exact same, comprehensive test. It'll just say updated. Everybody okay? Does everybody understand? Okay, thank you. Thank you for giving me some feedback. Okay, so that's the plan for the rest of the week. So that means we have 2 lectures today, 2 lectures this week, 2 lectures next week, and that will be it. All right, so unfortunately, there's some material on the last lecture that we won't get to, and I'm going to have some questions that will be on that material, but it'll be very general, dealing with cranial nerves, so at least you'll have some experience with that. Cranial nerves are a big deal, but I think that my questions will have you investigate them and at least be exposed to them. So you won't say, oh, we never got to that because Mike had surgery or something like that. So I want to make sure we still cover the material, and you'll still be tested on it, but it's all take-home. So I think that's doable. And remember the rules on take-home. You can use any source, computers, books, anything. You just can't use living people. You can't work on it in groups. You can't text me questions late at night saying, is this right? Now, you can text me a question about the take-home and saying, I need clarification. I've already had about 5 people do that. That's fine. But don't say, hey, Mike, will you read this over? No, that's a little.
Can I ask a question?
Yes, Ben?
The only, so the mitosis step, you want us to type out each stage and then underneath the typed heading, just
Are you talking
draw.
about drawing?
Yeah.
So for anything that includes drawing, just do it hand drawn.
Okay.
And just put it in as like an appendix or an attachment.
Okay.
Yeah, just attach it to the end.
Okay.
So type all your answers, but all the drawings you can do by hand.
Okay.
I would prefer hand drawn, even if you're a bad artist, versus cutting and pasting some picture from Google.
Okay.
Yeah, Dave?
On August 6th, you say there's an election, what's the time schedule gonna be?
That's a great question. So on August 6th, we'll probably do the same thing that we did for the 4th of July. The 4th of July, where we say we'll do like waves, 1 at 5 and 1 at 6:15 or something like that. Yeah, we'll do something like that. Yes, Joel?
Is the lab final comprehensive, or is it just
No.
on the last, it's on the last section?
Just the last section, yeah. Okay, so that means that will give me Tuesday and Wednesday to grade like a mother before I have this eye surgery. And then, hopefully I'll have your grades, this would be sweet, by Thursday morning. Now, I don't know, that may not happen. Reading some of your take-homes will be like 10 pages long, and for me to grade all of those in one day, it could be possible, but, we'll see. I'll try my best. Okay, so Omkar, since you're 15 minutes late or 30 minutes late, whatever you are, I put some stuff up here, and just ask your brother Michael to explain all that to you at some point. Okay, so, everybody happy? All right. Okay, so today we get to move into our last 4 lectures. So our lectures are going to be dealing with the nervous system and with this thing right here, which all of you should be familiar with. This is the cell body of a neuron. So I think that's good. You can see today we're going to talk about neurons, and then we're going to get into some electrophysiology, which sounds really scary, but it's basically just explaining action potentials again, which all of you should be okay with, like depolarization and repolarization. So today I'd say on a scale of 10, it's probably a good solid 6 in terms of degree of difficulty. I would say the sliding filament theory was up there close to an 8 or a 9. So not too tough of a day. Okay, so let's get into the overview. And one of the things about the nervous system that I think is important to mention is that it's one of 2 systems that's involved with communication. One of 2 systems that's involved with communication. The first thing that you're going to cover in 152 next semester or whenever you take it is the endocrine system, which means all about hormones. So hormones are a chemical that communicates. It's time to go through puberty, it's time to ovulate, it's time to grow more hair, it's time to make muscle, it's time to release milk. All these different hormones. So that's interesting. We'll start that next semester. But this semester, we're gonna end with the nervous system and its ability to communicate using chemicals called neurotransmitters. Neurotransmitters. Okay, so I'm gonna draw something on the board, and I think that this is a really important drawing. We're gonna continue to add stuff to this drawing as we go on through the lecture today. But when we think of the nervous system, we're gonna talk about the organization for a second. We're gonna say that it follows a very simple pathway. Simple. And it starts with a stimulus. The stimulus could be, wow, that's hot. Wow, that's bright. That hurts. I'm hungry. Whatever. Oh, your blood pressure is low. The stimulus can be external. The stimulus can be internal. In most cases with internal stimuli, you're not really made aware of it. It happens like behind the scenes. So things dealing with like blood calcium levels, we never know. When osteoblasts and osteoclasts are activated, we never know that. But those stimuli are picked up by something called a receptor. So a receptor identifies a stimulus. That's step one. Step two is that it's sent to the spinal cord, which we're all familiar with from lab. We know the spinal cord contains both white and gray matter. And the spinal cord is an information highway where we have information traveling up towards the brain and away from the brain. Okay, that's good. Once the stimulus gets to the spinal cord and goes to the brain, the information travels quickly down to something called the effector. So when we look at the steps, we're going to say that the first step is identifying some type of stimulus with a receptor. You may have learned in high school that we have lots of receptors, like photoreceptors for light, thermoreceptors for temperature. There's a lot of receptors. And when that information is sent towards the brain, we say it's sent in a directional term. We call it afferent. Afferent, which means towards something. When information is sent away from the spinal cord, in this case, we say it's efferent. These are directional terms. This might be easy to remember because efferent, right, is going towards the effector. That's easy to remember. Okay, so now we have directional terms. The next thing we need to add to this drawing is the word neuron. And I hope that we're all familiar with what a neuron looks like. We've drawn this in lab. We said it has dendrites, which are antennae. It usually has one long tail. And at the end of the tail, it has a knob, which we're sick of seeing in lab, that model, right? So we have dendrites. We have an axon. We have a cell body. And we have a knob. far, so good. We might add a few more things to this drawing as we get further into lecture. But the type of neuron that's involved with afferent transmission, which means taking something from the stimulus, from the site of the receptor towards the brain, we call this a sensory neuron. So a sensory neuron takes information in an afferent direction. Now you have to deal with some lingo. Hopefully your drawing will mimic this and you'll be able to make sense of this. Once we get into the spinal cord and the brain, we deal with another type of neuron, and these are called interneurons. Interneurons are in the brain and spinal cord. And once the response is generated by the brain, it's sent in an efferent direction using a third type of neuron called a motor neuron. So you may have heard of a motor response or some types of response. Yeah, Joel?
Do the interneurons go both ways?
Yes.
Okay,
Yes.
thank you.
So now we took a pretty simplistic pathway and we just added some labels to it. It's still simple. Stimulus, spinal cord, brain, effector, that's pretty simple. And we can say, look, this could be your fingers feeling something hot on the stove. This could be your eyeball saying, whoa, that's beautiful. This could be your arteries saying, whoa, you have too much blood in this pipe, high blood pressure. So whatever the stimuli is, it's following this pathway. In lab tonight, we're going to grab a reflex hammer and we're going to whack your partner's knee. And your leg's going to be like this, right? We all know what it's going to do. But when you whack your patellar tendon, listen, it's going to send information towards the brain in a sensory. It's going to go to the brain. It's going to have some type of motor response. And usually when we think of effectors, we're going to say that they're either muscles or glands. So in the case of the patellar reflex, the effector would be your quadriceps, causing your leg to go like this, to extend out. But I also like the fact that this pathway involves reflexes that result in glandular secretion. You can ask my wife. I use this phrase probably once every 2 weeks. If I'm really hungry and my wife is making something really good, or she suggests that we go to someplace that that has really great ribs or something like that, or really good ice cream or something like that, I will say to her, just if she mentions that, I'll say to her right away, I'm starting to squirt spit right now thinking about it. And she'll say, what? And then I have to tell her, when I smell something, I'm anticipating the food and I'm getting my saliva glands ready. I'm starting to squirt spit. So now my kids know that, man, when that's getting me going food-wise, my saliva glands are hyperactive. Works with dogs too, right? My dogs are standing there looking at me while I'm grilling, waiting for old Mike to throw him a piece of hot dog or hamburger. My dogs are like dripping spit on the ground, just saying, please, please, I'm begging. I can smell it, I'm anticipating, right? So this pathway, we can describe it as the basic pathway of the nervous system. We can even describe this pathway as being involved with, ready, reflexes, which is a topic for today's lab. So this is a money pathway, which means you're going to have to, ready, hand draw this and explain this on the final take-home exam. It's kind of sad, isn't it? No more sitting down in here and coloring in bubbles.
So sad.
So sad, so sad. All right, so there we go. There's the introduction to the world of the pathways that we're going to be talking about for the next 2 weeks. All right, so let's talk about the organization. When we say the word nervous system is divided into the central nervous system and the peripheral nervous system. Central. Central means brain and spinal cord, and peripheral deals with all of the nerves. So that should be easy. Brain and spinal cord, central, nerves, peripheral. Okay, so far so good. So let's take a look at this diagram. This does not do the peripheral system justice, but you can see that you have the brain and spinal cord. So remember last week when we talked about the spinal cord being surrounded by meninges? We talked about that one tough mother. Well, today in lab, we're going to be dissecting the brain. And the brain is surrounded by one tough mother. And you're going to pull it apart, and you're going to say, whoa, there's the arachnoid, I see spider webs. Oh, the brain is shiny, that's the pia, just like the spinal cord. So the spinal cord and brain are one kind of subdivision. The peripheral system has all of these nerves, and there's way more than this. I mean, the nerves are amazingly complicated. And what is literally mind-boggling to me is that when these nerves are sending information towards the brain in an afferent direction and then off to the brain can identify exactly where that stimuli came from instantly. Instantly. I mean, if there's a fly flying into your eye, you blink. Think about how fast that stimulus comes in your response. I mean, it's less than a second. So it's almost like it's impossible, right? Like, how can that really be? So we're going to talk about the whole process of identifying and responding, but we have to use some lingo. We use these terms already on our diagrams, sensory and afferent. Those go hand in hand. That means towards the central nervous system. Motor and efferent, going towards the effector. So that's good. We're good. You can see when we look at that the motor response, it can be visceral or it can be somatic. Now I want you to write those two words down. Visceral and somatic. Visceral is usually something that you cannot control voluntarily. It's a visceral response, right? Meaning that it's involuntary. So like digestive things, blood pressure, so many things that we are not aware of. If the response is an effector that's somatic, it usually means skeletal muscle. So as I wrote on the board, the effector can be either muscle or glands. And that's what we see here, visceral or somatic. Now take a picture of this. This probably won't be on the exam, but you may have to explain this on the exam. I'm gonna have to be really careful about what I say is on the exam now, because it's only like 12 written questions. So one of the things that I want you to feel confident with is kind of explaining this organization. So let's do it together. Ready? We said that the that the nervous system consists of two major subdivisions. Stay with me, just follow me for a minute. Central, which is brain and spinal cord, and peripheral, we said all the nerves. But we know that some of the nerves are coming towards the brain and spinal cord. Where are they coming from? Look, where are they coming from? Well, some of them are coming from stimuli from like your digestive system. We call those visceral sensory. Some of the stimuli is being identified from skeletal muscle, like, whoa, I'm exercising, I'm moving, I'm doing this. So this information is coming towards sensory, whether it's visceral or somatic, remember, involuntary or voluntary. And then once the sensory information takes it to the brain and spinal cord, a response is generated. So the other set of nerves are motor responses. And we said that the motor responses can be somatic, like whacking your knee gives us our quadriceps to move our leg. We can see that. But if the response is visceral, like behind the scenes, involuntary, You know, it's hard to see a visceral response. Sometimes you can say, oh, I notice my heart rate is increasing. Oh, I notice I'm breathing faster. Those are things that are visceral. But notice how the visceral response is subdivided. And put big, big stars next to this word. Autonomic. Say it again. Auto, like car, nomic. N-O-M-I-C. Autonomic. When you hear the word autonomic nervous system, you're going to say that it involves, look, a visceral motor response, which means a response that we're not aware of or we can't really control. So an autonomic visceral response can either be sympathetic or parasympathetic. And whoever you have for 152 is going to be teaching a lot about these two terms. So I want to make sure that you guys don't forget this. Sympathetic is a response that gets you ready to fight. What's that mean? Get your blood pressure up. You're starting to breathe a little faster. Your heart rate's starting to pump faster. Your eyes are dilating. You're ready to go. So you can say, I have lots of examples of a sympathetic response. You can say, someone just took my kid at the mall. What are you going to do? Oh, you're going to get into father or mother bear, and you're going to go get them no matter what. I mean, look at that. I'm getting goosebumps just talking about it, right? You're going to get fired up to do it. Or you can think of like the Olympics. That's what I think of. Like doing like the 100-yard, 100-meter dash. It's a sprint that lasts what? Less than 10 seconds? And these people have trained years, maybe 10, 15 years, for a race that lasts less than 10 seconds. Can you imagine what their heart is doing when that dude says, on your mark, get set? What? 10 years of training comes down to this. I mean, if you're not pumped up and like totally amped sympathetically, then you're not going to do well. But after you do whatever, your body needs to recover. And that's what parasympathetic is. So you can write down that with sympathetic, it's getting ready to fight. Parasympathetic is about resting and digesting. Ooh, resting and digesting. If any of you do something really intense for like 5 or 10 minutes, the last thing you're going to say is, oh, I got to pee. No. Your body says we'll save that for later. Or your body's going to say, I'm not going to worry about digesting food. We got to get energy to do this amazing event. So both of these are a response motor, but it's going to a visceral, remember what that means? Something that we're unaware of or involuntary. So now when you look at this diagram, which looks kind of easy, you should be able to say, okay, brain and spinal cord, check. Nerves, check. Oh, they can be sensory, they're coming from these two, or they're going to these two. If the response is going to something that I'm unaware of, it can either get me ready to fight, or it can get me ready to rest and digest. Yeah, David.
All right, I got to figure out how to phrase this. Earlier last week, we were talking about people hitting the wall because
Yes.
they burned up their bodies and immediate energy.
Yeah.
So they go into I guess they would go into a parasympathetic.
Yeah, so parasympathetic would be helping kind of recover. Yeah.
Now the people who push through, the people who push through it,
Mhm.
are they still relying on the parasympathetic, or are they kind of like?
Well, remember, so that had to do with like strenuous exercise. Usually a sympathetic response is a burst. So it's just like an adrenaline rush. I doubt that someone who is running a marathon for 4 hours is always amped up. I think they're kind of getting to a groove. In fact, really people who run a marathon don't even breathe hard. Even after like 3 hours, they're so in shape. It's just that had to do more with fuel and not so much an adrenaline rush where you're going crazy. So think of these responses as getting you ready to do something amazing, but only for a brief amount of time. Okay, in our figure that we drew on the board, we used these terms. We said that there are sensory and there are inter and they are motor. Notice that it says up here that interneurons are the most common of all the neurons. Okay, so far so good. Nothing too tough. Here's the diagram that we drew on the board, but with pictures. Here it says, whoa, I'm putting my finger on something sharp. So there are receptors identifying that pain. It's traveling, look, towards the brain and spinal cord through sensory and afferent. It gets to the spinal cord, goes to the brain through inter, and then the response is, hey, move that finger off of the stove or away from that sharp object. That would be the effector. Okay, so notice that they organized this pathway, which we drew on the board, into central, which is brain and spinal cord, versus peripheral, which is sensory and motor. Okay. Now, when we look at neurons, we said that they share some characteristics with muscles. Muscles are excitable. Yes. Irritable. If I said to you, explain to me why muscles are excitable, hopefully you would talk about the membrane being kind of somewhere in between a positive and a negative environment. Same thing with nerves. Muscles are conductive, which means heart muscle can transmit electricity very quickly so the heart beats in a rhythm. Nerve cells are also conductive, obviously. But here's something that muscle cells are not, or unable to do, and that is secrete. So muscle cells can release nervous tissue, neurons can secrete neurotransmitters to do their communicating. So 2 out of the 3 are shared with muscles. Secretion kind of separates or makes it special. Only nerve cells, neurons can do that. So we drew a neuron over on the board, and I think I drew a lot of these things. Dendrites, the cell body with the nucleus. We drew a long axon with a knob on the bottom. So far, so good. What I did not draw on our drawing, which you can draw, is some rectangles. So I would expect the rectangles to be on your drawing. I don't think I need to go draw those. But the rectangles on the axon represent the myelin, the insulative wrapping. And just like we said in lab, that myelin, this insulative wrapping, is incomplete. They have these gaps or spaces called nodes of Ranvier, which was kind of part of our lab last week, right? So nodes of Ranvier represent the axon not covered by myelin. The other thing that I want you to write down is this word trigger zone. That's one of my favorite terms of the neuron. So the trigger zone is kind of like this area where the cell body kind of forms the axon. We'll talk more about the trigger zone, I think, on Thursday. But you can say now in your notes that the trigger zone is where a lot of the decision making is made, whether or not a neuron is going to respond or inhibit some type of response. We'll talk more about that on Thursday. Okay, so everybody's good with this diagram and the figure that I drew on the board. You should be able to redraw them. You do not have to know anything about this, so just listen and just relax. But it says that we have different shapes and sizes of neurons, and it's based on how many things are coming off the cell body. Now, I believe on the exam there was one multiple choice question dealing with this, but you're never going to see a multiple choice question from me again, so this is not going to be on the test, right? So the word multi, bi, and uni says that when we look at this particular axon, this cell body, it says there are a lot of things coming off this cell body. Many multi-dendrites. Bi means, look, we have only 2 things coming off. We have an axon and one of these big branches. Unipolar means we only have one branch. And if we have an and, now remember this prefix in the world of anatomy, and means without. If we have an and axonic, which means without an axon, then we have this weird situation where we have a cell body with just dendrites, no axons. So multi means more than one, bi means two, let's say more than two. Bi means two, uni one. Okay, you get it. All right. Now, one of the cool things about a large cell like a neuron is that the knob is just as important as the cell body. And we have axons that travel from your butt cheeks, think about that, down to your ankle. That's one long axon. So how in the world are we able to keep the neuron alive, both the knob and the cell body? Recycling, making sure that everything is taken care of. We have transport. So transport up and down this axon is either anterograde, which means either direction, or we have what we call retrograde, which is going only backwards towards the cell body. And one of the interesting findings from this idea of transport of organelles and proteins and things like that is that when we look at retrograde, we say now that that's a possible, think about this makes sense, everyone think about this. It's a possible place where we can incorporate something foreign into the nerve cell. How do you do that? Do you remember what happens when the knob is done with excreting that neurotransmitter? Endocytosis. Remember? Guess what? Virus can hop on and get sucked in. And then they're transported back to the cell body and then they can make that cell sick. So we think now that retrograde is one way or explanation on how some cells get sick by having that virus or bacteria hop aboard with acetylcholine or whatever neurotransmitter and get sucked back into the knob. In lab, we talked about helper cells. And you know what? I like to draw so much, I think I'm going to include this. So when we have our axon, I said please include the myelin. See, I have to do this. So there's our myelin, and the gaps are called nodes of Ranvier. Then I believe in lab, what I did is I had you draw some little stars all the way around your neuron. And what I tried to make these stars represent were neuroglial cells. Neuroglial cells. And I would say in a third grade anatomy class that neuroglial cells are helper cells for neurons. But they have lots of different functions and lots of different names. So you know that on the lab practical, which is on Tuesday, August 6th, that you're probably going to have to look at that figure from last week's lab and identify the neuroglial cell by name and give me one sentence explanation. So if you haven't done that, you're in good shape because we're going to talk about the functions of them now. First one we're going to talk about are neuroglial cells called oligodendrocytes. What a name. And its main function is to wrap the axons of central nervous system axons only, not peripheral. Wrap them with myelin. So oligodendrocytes utilize myelin and cover the axons only in the brain and spinal cord. Epidermal cells are cells that are responsible for producing, and here we go, look up here, cerebrospinal fluid, CSF, cerebrospinal fluid, which is the fluid that flows in between the meninges. Some of you are saying, oh yeah, the arachnoid layer. It's also fluid that circulates around the spinal cord. Some of you are saying, oh, spinal taps, right? So thank you, epidermal cells, for making that fluid. Microglial cells, this is kind of fun. I just talked about these at Xavier this morning, and I'm teaching the second semester at Xavier right now. And you might say, well, wait a minute, I'm taking the second semester in the fall. So that means probably you're going to hear about microglial cells again. So that's something that says that it could be important. Microglial cells are immune cells of the brain. Microglial cells are cells that are usually found in high numbers when there's some type of, and underline this in your notes, ready? Lesion, L-E-S-I-O-N. You will be a college-educated smart nurse, or whatever you want to be, if you remember that a lesion is some type of damage to the brain. Damage could be a stroke, it could be a tumor, it could be infection, it could even be Alzheimer's. And in any of those situations, guess what? You're going to find a high number of microglial cells around that lesion. Sometimes people will die with dementia. And in order to verify, sometimes people will say, yeah, my great-grandfather had Alzheimer's. He would want his brain investigated and researched. So they will slice the brain in very thin cross sections. They'll look underneath the microscope, and they will see a tremendous amount of microglial cells, which will help support that. That, oh my gosh, you didn't have a tumor, but there were microglial cells everywhere.
Do those just form, or they're like just there?
No, they're always there. But in terms of battling, they can call out and bring more in from other areas of the body.
So it's supposed to help heal the trauma, or?
Yes, so microglial cells are a form of white blood cell called a monocyte. And a monocyte is usually like the big engulfer of foreign bad things. So yeah.
I think that's common with football players, CTE.
With like CTE, probably, I would say so. People who have
That's trauma.
repetitive concussions probably have lesions, probably. Yeah, we'll talk about CTE. Okay, so here's a diagram that looks similar. I wouldn't take a picture of this, but similar to the one in the lab manual. Make sure you know the picture of the lab manual. So here's the cell body. You can see some epidermal cells. You can see some microglial cells, oligodendrocytes. Remember, you just have to be able to take the picture that's in the book and identify them. There's a couple more neuroglial cells, so let's talk about those. One is called an astrocyte, and the other two are called Schwann cells and satellite cells. Now, astrocytes are the most numerous. That's probably something that's important. And they're involved with the BBB. That's a big term in brain world. It's called the blood-brain barrier. So in most cases, things that are in your blood that are dangerous are unable to pass through this blood-brain barrier. But it's allowing things like glucose and oxygen and things like that to get through. But there's always an exception. For example, anesthesia. You know, if you're having surgery, I'm sure with my surgery, at least I will be in somewhat of a la-la land, I think. I might even be completely out. I want that anesthetic to penetrate my blood-brain barrier so I do not feel stuff going on in my eyes. You know? When you think of blood-brain barrier, also you can think of alcohol. Alcohol can penetrate that like wildfire. So that's a flaw in the design. So people who drink excessively have slower responses. We'll talk all about that. So those are the only things that I want you to know for my test. is the top 2 bullet points. You can see that there's other things about astrocytes, but just remember that they're the most numerous and they contribute to the blood-brain barrier. We said oligodendrocytes work with myelinating the central nervous system. Well, what about all those peripheral sensory and motor nerves? Well, look, Schwann cells. So that's something that you don't want to confuse. Schwann cells are peripheral, oligodendrocytes are central. They both do the same thing. And then satellite cells. Can you believe this? In 2019, we still are unsure about what some neuroglial cells do. It's kind of cool. All right, so when we think of neuroglial cells, I want you to think of this incredible kind of matrix. All of these big, kind of looks like hairs or axons. They're all sending signals, and we have all these support cells all the way around it. And this is probably just, 1/1,000,000th of a centimeter. It's probably just a micrometer in size. So we have many, many neuroglial cells. And that's a problem, because when we think of neuroglial cells and what happens to them potentially can be very catastrophic. We'll talk about neuroglial cancers in a minute. Let's talk about this stuff called myelin, since we've talked about oligodendrocytes and Schwann cells. When we look at myelin, we're going to say that myelin is an insulative wrapping, but here's something that I want you to underline in your notes. It's mainly fat. And look what it says next to it. It's 80% lipid. It looks white. So when we talk about white matter and gray matter, this is a potential NCLEX question, I'm sure. When we talk about white and gray matter of the brain, you're going to say white matter has myelin. And the only structures that have myelin are axons. So that's really easy to remember. White matter has axons, the long tails. Gray matter are parts of the neurons that do not have axons. That do not have white matter. So that would be dendrites, cell bodies, the synapse or the knob. So remember, white matter is white because of lipid. When I also see this word lipid, I also think back to when my kids were really young. And my wife was and is a school teacher, and when she gave birth, she breastfed for like 4 or 5 months, I think. And then we transitioned into formula for, I can't remember how long, 3 or 4 months. And then we transitioned into Kroger milk, or UDF milk, wherever you buy your milk. And I can remember the pediatrician telling us that when we buy milk for the first time for our kid, this is going from powder formula, he said to always buy red cap milk. And we looked at him like, we are blue cap in our house. Skim milk, we're trying to watch our weight. Well, when we think of the process of myelination, it needs the ingredients of fat. Fat is a big part of breast milk. And so when you give your baby who's a year old or 6 months old real milk, you want to make sure there's enough fat so that the nervous system can continue to develop. Now, how important is that? That's awesome. That's like taking anatomy and saying, oh my gosh, this is real world stuff. Because someday you may have kids, or you had kids. You might remember this, and now it makes sense. Jocelyn.
Is that why they say that fat babies are always the smart babies?
Oh, I don't know about that. My daughter was pretty down. That's mean, Joe. My daughter was very chunky. She's very smart. She's very smart. She's going to be a great kindergarten teacher. She's very smart. I said that 5 times. No one's going to put that on social media. Someone else had their hand up. Oh, Caleb, did you have a question?
Yeah, I remember with my son, they wanted us to put him on the whole milk because they said for brain development. so
Yes,
basically-
it's all about myelin. Yay. Good.
Fascinating.
It is, isn't it?
How does DHA play into that? That's a big thing now.
Yeah, so DHA is an amino acid form, and that's also helps with protein building.
So it's just a different form of brain development.
Yeah, so that helps with they think maybe like dendrite formation. That's kind of cool, yeah. All right, so we're good with myelin. We know that there are gaps called the nodes of Ranvier. We said that the initial part where the transition from the cell body into the axon is called the trigger zone. Where the signals begin, but it's also where the decision-making starts as well. We'll talk more about that. So take a look. If I would say, put this on the test and say, hey, is this axon from the central nervous system or the peripheral nervous system? You would say, whoa, Schwann cell, that's only peripheral, right? So this is either a motor or a sensory neuron, not an interneuron, because Schwann cells only do peripheral. When we look at Schwann cells and the way they wrap, look at this. It starts out maybe in the womb, beginning the process, but it's like a jelly roll. It continues to wrap, 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 wrap. And so this is the process that continues. Tara, can I grab your foot for a second? Show me flexibility. All right, feel the burn. Ready? So if I took, give me your pen. So if I took Tara's shoe off and sock off, and I'm not, and I took something sharp and I stroked her foot, starting at her heel, going up to her big toe, her toes would go like this. Oh, that tickles. Like, watch. Oh, that tickles. But if you do this to a newborn baby, and you start from the heel, you go up slowly towards the big toe, the toes go like this. Ah, like that. What? Or if some adult is in a car wreck where they can't talk, you know, you think that they're unconscious, they've had facial trauma, you can do this where you can stroke the bottom of their foot to see what kind of response. If an adult gives you a baby's response doing this, then that means that part of that pathway has been altered or destroyed. So one of the things that you think about, at least I did when I was a dad of early age kids, is that I'll never forget taking my son Zach, who just got married 2 weeks ago, it's so weird, to his first pediatrician trip. It was like a week after they were born. And I went with my wife, but I was interested to see like, what does this pediatrician gonna do? And the pediatrician was like holding Zach like this, and then not gonna drop him, but like acted kind of like he was gonna drop him, and the baby did all these things, and then he went like this, and he said, "Hi, Zach," and he put his finger right here. And if you've ever done this to a baby, the baby instantly grabs your finger, and you say, "Oh, she likes me the best. Look, we are bonding. Look, she's grabbing my finger." No, it's a reflex, right? Because notice Tara's like, "Mike, get off of me." Like, I do not want to touch your finger, Mike. But if Tara was a baby, she'd go like this. We would be bonding, So the reason why we get weird responses in infants is because we're at this stage. Look, the myelination isn't complete. So that's why moms are encouraged to get more fat into their baby's diet up until about age 4 or 5 until myelination is complete. So if you don't leave, if you leave today and you say, "Oh, this was a dumb lecture," that's pretty cool, I think. It's pretty interesting. You can also see that myelin can be a source of, horrible disease. Look at this. This is normal myelin, the nodes of Ranvier. And here is myelin that's been destroyed. Look at this, by your body's immune system. So this eventually affects how fast information travels from one point to another. So people with multiple sclerosis have myelin that's being destroyed. For some reason, the immune system is targeting myelin as being a bad thing. Oh my gosh, how horrible. We said oligodendrocytes do it in the central nervous system, and again, it's 80% white. I think this is the same slide. Yeah, so same slides for some reason. All right, so here's what an oligodendrocyte looks like. You can see it kind of looks like an alien with arms that are wrapping around the axon. similar to a Schwann cell. Now, this is a pretty cool slide because it says in the world of the nervous system, bigger is better. When we look at the world of how fast information travels, we're going to say bigger and myelinated are the fastest. So a large myelinated axon is the fastest of all axons. But notice that there are some axons that are small and unmyelinated. Now, if you and I were the grand designer of a human, I think we would say, let's have every axon being gigantic with tons of myelin, so it responds instantly. But we need places where we have a slow, listen, and steady response. So in your notes, next to the word unmyelinated, put the word stomach down. Because your stomach has to have a slow and steady response in terms of producing stomach acid. Because it never knows when all of a sudden you're going to grab a handful of M&Ms or drink something. And your stomach can't be dry. I mean, come on. It has to have something in it. And of course, when something gets in the stomach, then you send stimuli and your brain makes even more. But until that happens, until you ingest something, your stomach is always producing a slow and steady amount of stomach juice, gastric juice. And you could say, thank you, small unmyelinated fibers, for continuing that production.
Where
You
would
with
it be
me?
large at?
Oh my gosh, large would be supplying big muscles, large would be supplying information to the heart, things like that. Yeah. Okay, now this will not be on the exam, but I just want to briefly mention that we defined nervous tissue back when, in the second week of May, as being a type of cell that was unable to undergo mitosis. That was one of like the characteristics of nervous tissue. Can't repair, can't divide. Well, here you can see that sometimes in weird situations, it can heal itself. So it depends upon where the damage is on the axon. And if you are, thinking, well, how does, Ryan Shazier, if you're into football, who was on the Steelers and got paralyzed, couldn't walk, and now he's walking again. How does somebody go from being paralyzed to walking again when nervous tissue doesn't repair? It probably means that this part of the neuron was able to regenerate. So it's possible. So remember that. Okay, now we get into the electrophysiology. I think we can go somewhat quickly through this. Most of you who are here today, I think almost all of you either got a C+ or higher on the test, so that means you kind of know what's going on. The word polarized means that we have an imbalance in electrical current. So we know that in muscle cells, remember what we called the cell membrane, the sarcolemma? We said that on the inside it was much more negative, on the outside it was much more positive. It's exactly the same. It's exactly the same in a nerve cell. So there's an unequal balance of sodium and potassium and chloride on the outside. Remember, ECF is the outside extracellular fluid. ICF is the inside intracellular fluid. And we know that cations and anions create an electrical imbalance, making the nerve membrane on edge. We call that resting membrane potential. So sodium and potassium come into play, which means we have sodium and potassium channels. That means, and you don't have to study all of why we have negativity, but we know that there's going to be shifts. So here